Can you see my screen? Can you see my screen? Hello?
Yes, we can. Yes.
Yes, we can. Sorry,
Okay.
I was on mute.
Thank you. And it's been already recorded, right? Okay, thank you so much. Hello, everyone. Thank you for joining the IUPAC WorldFAIR Chemistry virtual webinar series. This is our second webinar in our series. It's a short series that is mainly made to discuss what is a chemical from a data perspective. This time we are trying to answer a few questions about what is a chemical across applied chemical areas. We have experts and amazing speakers, as you can see here, from different applied areas. Thank you all of our speakers for accepting our invitation and joining us. And thank you for our audience as well for joining from across the world. So let me briefly here go through the webinar program for today. So after short discussion or short introduction, we will be having flash talks for 5 minutes of each speaker to talk about how to handle chemical data in their areas. As you can see here, we have topics such as artificial intelligence and machine learning, agrochemistry, textile and dyes, and pharmaceutical and chem informatics. After that, we will be having a panel discussion. So if you have any questions, please feel free to write it in the chat box as well. If you want to share your notes with the whole community who are attending this webinar, please go ahead. There is like a link in the chat box. You can also get engaged with the audience. After that, sorry, I don't. Okay, after that, we will be closing with a few announcements, so stay tuned until then. All right, so let me get started here with a little bit of introduction about the IUPAC. IUPAC is the International Union of Pure and Applied Chemistry. It's the worldwide authority on chemical nomenclature, terminology, and symbols. So if there is any new naming for, let's say, some elements in the periodic tables of standard methods or, you know, chemical data, chemical data, IUPAC is the authority on that. The IUPAC is not only doing this, but it's also engaged in many other activities to advance the chemical profession and to engage with the broader community in the whole entire world. So they are working on, for example, publication journals, books, databases, and technical reports. They are working on also recognizing scientific contribution, as well as building worldwide capacity by providing trainings and education and outreach, and many other projects. The mechanism of work in the IUPAC is based on projects. So projects are usually proposed, and then when it's got approved, committees and divisions start working on that. As most of you probably know, and we have a huge number of volunteers who are doing this amazing work. So another thing that the IUPAC is engaged with is the digital, we call it the digital IUPAC. As you can see here, the IUPAC is engaged in a network through collaboration with the broader chemistry data community to translate communicating standards into digital domains. It's based on the 3 main pillars, as you can see here: the chemical representation and notations of chemical substances, the chemical concept definition as one of the projects we have been working on is, for example, the Gold Book, and the chemical properties of, let's say, critically evaluated data in general. So the main goal here for the IUPAC is to develop machine processable technical description, building the IUPAC authoritative scientific definitions. And thank you again for the collaborators who made this work happen. Thank you for all of our collaborators, as you can see here, InsureTrust, NFTI for Chem, PSDI, PubChem, GoFair, GoData, and RDA as well, who've been engaged in this effort. One of the projects that So I'll talk about this project later, but I'll mention here, as this webinar is brought to you by the IUPAC and the WorldFAIR. What is the WorldFAIR as well? So the WorldFAIR project is a new global initiative mainly made to advance the implementation of the FAIR data. When we talk about FAIR, FAIR means findable, accessible, interoperable, reusable data principle. So as you can see here in this right corner, the WorldFAIR is engaging different, I think 11 cases to have this conversation about how to implement FAIR data across disciplines. Chemistry is one of those disciplines. The chemistry case is led by the IUPAC. And this is good here to mention as well that this work has been done with coordinating with the CODATA and the Research Data Alliance RDA. For our project, the WorldFAIR Chemistry, this project is led by the CPC EDS committee of the IUPAC, which is led by Leah McKeown. We have created 3 subcommittees or subgroups who are working tirelessly on or accomplishing the deliverable of this project. The first committee is the D31 committee, who are working on reporting guidance. The main goal of this committee is to prepare guidance for implementation of FAIR data principles, which could benefit different stakeholders like researchers, publishers, funders, and so on and so on. The second project or subcommittee is the training cookbook, the D3.2 committee, who are working on preparing and developing a cookbook, like a recipe cookbook for depositing FAIR machine-enabled chemical data and identifying and extracting chemically relevant metadata. The third group is the T3.3 group, who are working on developing a protocol services. Think about browser-based services that enable real-time feedback for verification of machine-readable for everyday chemical data and metadata representation. Think about a global chemical resolver and chemical structure validity checker. So these are the 3 main groups that we have in this project, and they are working very tirelessly to have this job accomplished. Thank you for everyone who has been working on that, and we're happy as well to have more volunteers. And thank you for our collaborators that I mentioned earlier who have made this get started. All right, so in our today's webinar, as I mentioned, this is our second webinar in this series. And what we are trying to answer here, to collect some answers and thoughts and opinions from our speakers about the chemical substances and notations and chemical data handling across their areas, about characterizing chemical data, chemical substances, chemical data in general application, what are the resources are used to analyze chemical data, and what are the challenges that usually faced by daily life, and how could FAIR data principles implemented, and what is the role of IUPAC here, and how we could have this work being sustainable and consider sustainability for data resources. So with that, I'll be introducing our moderator for today, Dr. Ian Bruno, who is actually leading the D3.1 committee here, who are working in the guidelines to moderate this session. So Ian got his bachelor degree in chemistry from Durham University and a PhD. That time it was called Information Science from University of Sheffield. Since then, he has been working in Cambridge Crystallographic Data Center, CCDC, for about 30 years now. So he is the scientific, he has contributed to the development of many core softwares products, most notable Conquest and Mogul. Now he is working as a director of the Data Initiative, responsible for establishing a strategy that will make CCD data more readily discoverable and reusable by wider scientific community. He is also engaged in many global initiatives such as the IUPAC, and he is also the secretary of the NSHE Trust. Without further ado, Ian, Bruno.
Thank you very much, Fatima, and thank you for coordinating this event and bringing together a set of speakers that I think is going to give us hopefully quite a broad perspective of what is a chemical across a number of industries. Fatima mentioned my many years at the CCDC. A lot of our interaction with the real world application of our data, I think it would be fair to say, has been in the areas of drug discovery, and we do have that area represented here, but it's good to see other aspects of chemical industry represented too. We're going to start with just a brief poll to sort of just get a flavour for who's attending this particular webinar. If you came to the first one, some of these questions will be similar, but some of them will be different. And so the poll's being displayed. I think you can basically go ahead and fill it in at your leisure. There are 4 questions in total. One is just asking about your background in terms of whether you're coming from academia, industry, or other areas supporting the chemical enterprise. The second is getting an assessment of how important machine readability is to your work. The third, how easy it is. And then the final one just gets your perspective on how aware you are of FAIR data principles, and we'll be assimilating these results and looking at them across the sort of like spectrum, just to get a flavour of who's coming to these particular webinars. So hopefully I can leave that running and move over to introduce our first speaker, which is Lutz Weber. Lutz, if you'd like to start sharing your slides. Lutz has more than 20 years practical experience across pharmaceutical chemistry, drug discovery and development, and has been responsible for identifying new drug candidates and moving projects into clinical development. He's also a co-founder of a number of biotechs in Germany and the US, and I've certainly been aware of work that he's been doing through Ontochems with semantic relationships, Google and BigQuery, which has been pretty impressive. So looking forward to what Lutz, you have to say about what a chemical is from your perspective. So Lutz, over to you.
Yeah, thanks Ian for the introduction. And I would like to speak here. I'm happy to be the first speaker, of course, to speak about what we are doing in the field of chemical data and how to use chemical data in our context, saying that Ontochem is really a chemoinformatics company, and we are dealing with a lot of chemical and other data. And so basically, when I was a student, we still collected data from small paper sheets like this and tried to do some SAR, structure activity relationship, and structure property relationship modeling using data written on paper sheets. Today, this situation has really changed dramatically because the amount of available data has increased exponentially. So we ourselves have access to more than 600 million scientific documents, which we extract and process. And basically, we annotate those documents with chemical named entities, with other entities like diseases, magnitudes, industries, and things like that. And this extracted data is basically terabyte scale. And in order to deal with that, we need some specific technology. And for this, we are using Google's BigQuery tables to store these data extracted from these 600 million documents. So these documents are like chemical patents, scientific full text publications, conference reports, scientific grant applications, and so forth. I would like to show you shortly one system for prediction of any molecular property, means using chemical ontologies as feature spaces for machine learning. And the example should be likelihood of drug approval. So if we could if we could predict the approval rate of a compound, this would be very nice. So ontologies are basically a representation of our knowledge. And what I'm showing you here is a section from our chemical ontology. So we have been active in the field of chemical ontologies. And we also did a automated compound classification system, which puts compounds into these compound classes. This is available for free at GitHub, so you can download the software and run it yourself and determine what kind of ice you are dealing with for the compounds. In the same way, we have also a range of other ontologies. This is basically a toxicology ontology, where we have a machine-readable representation of our knowledge about toxicity. So, for example, we have a class cytotoxicity, which is a child of the general concept toxicity. Now, using these ontologies, you can annotate the documents. And what I'm showing you here is one article which has a sentence in it, which basically contains toxicity data, because the sentence says this PM genotype has furthermore been associated with an increased risk of toxicity and metoprolol, for example. So this gives a co-occurrence with a chemical compounds and the toxicity term. And then you can extract relationships of these two named entities and put them into data sheets or data tables. And we did this as a showcase for all PubMed Central documents, where we have like 1.3 million compounds identified, about 6,000 compound classes, 816 toxicity concepts. And this basically gives rise to a structure activity relationship table with 1.8 million relationships between compounds and toxicity terms. This can be stored and retrieved in table format. And it could be also put into BigQuery tables. And what I'm showing you here is basically our way of doing FAIR data. We have a project there, which is called SciWalker Open Data, where we register chemical compounds. And what I'm showing you here is the registration database. It has currently about 158 million unique chemical compounds registered. We also have uploaded here different other databases like the IUPAC tox, the FDA compounds, universal natural product database, SAVI database, and things like that. So this is open to everybody. Everybody can go there and perform SQL queries on these compounds, download the data, and reuse it as it makes sense. Now, in our case here, very shortly, we have basically connected the chemical structure, the chemical fragments or labels, as we call it, together with the found toxicities. And what you get at the end is a prediction system where you apply different machine learning models on these data sets in order to predict a certain toxicity of a compound. And using that, you can basically generate something which we call toxicity score and use that as a classificator to predict clinical tox outcome. And basically, if you are in this blue range here, it would be rather safe. If you're going more to the right, it becomes more and more unsafe. And this could be used to predict toxicity, but it could be also used to predict approval rates. So if you're here in this region of 1, then it's highly likely that your compound could be approved or will be approved, and increases the likelihood of success for your compound. So that was a demonstration, my 5 minutes, and happy to discuss further if anybody is interested.
Brilliant. Thank you very much, Lutz, indeed. And what we're going to do is we're going to hold questions over for after we've heard all of us talks for when we have the panel discussion. If you do have questions that you want to throw out immediately, though, do feel free to use the chat to put those in there. And I think, you know, what I think I like there about what you demonstrated there, Lutz, was the sort of diversity of sources where we might be wanting to find chemistry information and the sort of complexity of the relationships and the challenges and opportunities that presents. So thank you for that introduction. We're going to move on now to Theo Leno, who comes to us from IBM Research. We haven't tried sharing his slides yet, so this is going to be an adventure. But while we do that, Theo is a distinguished research scientist and manager at IBM, developing machine learning and AI methods to digitalize chemistry. And this has led to IBM's RxNIAI tool, which supports various aspects of reactions and synthesis. He has a background in theoretical and computational chemistry, gained in Italy, now in Zurich, and now going to give us his perspective on what is a chemical. So Theo, over to you. We can see your slides, not in presentation mode though.
Yeah, no worries. Hopefully they will come soon.
Yeah, awesome.
But no. Yeah, cool. So we rely on improvisation. And we'll try how that works. Okay, so thank you so much, Ian, for the nice introduction, and also for you and the organizers for having me here. And I'll keep it really relatively short. I mean, within 5 minutes, to give people an idea on how we really go for having high quality data in the reaction chemical space, and how we use that one for then enabling people in downstream applications. So it's a very important thing. So this is of course, I mean, very short slide to thank all the people that have been contributing. I'm here talking about lots of young people excited to train machine learning model with chemical data. And I think I'll spend a few words here just to say that I'm a strong supporter of the fact that machine learning per se is actually creating that wave, that revolution that is making chemistry as a discipline way closer to young generation than probably we have been experiencing in the last 15 years. So it's definitely something very, very good, very interesting. It's bringing chemistry closer to high tech. So I'll skip a little bit of general information, and again, just apologize for going very, very, very fast, but the purpose here is really to give you a very quick understanding of how we use the data. And when we talk about data, I'm referring to chemical reaction. So for us, it's very important to have a standardized way of representing chemical reaction. We heard from the previous talk actually that the importance of ontologies and the importance of having things very well organized. And here is really not less important, although instead of working with ontologies, we really work with standardization and standardization languages. And it's important to have everything very well captured. So for instance, when we train predictive models that are capable of helping people to understand what are the outcomes of chemical reaction, we really rely on learning all the basics and the fundamentals of chemistry directly from the data. And so this is an example of how we use chemical data, and in this case, extracted from patents. We actually have a chemical reaction that is represented in a schematical way, in a diagrammatic way. This is very hard to be processed by computers. We had at the beginning of this seminar computer accessibility, a question about computer accessibility of the data. And this is an example in the space of synthetic organic chemistry where this information, although heavily present, maybe even in a fair way, even findable and accessible, reusable, it's very hard to be processed by computers. There is lots of image processing that is required, and there is lots of obstacles. Instead, we tend to rely on an equivalent representation, which is linear, that relies on use of tokens, characters, and strings and symbols. In this specific case, it's a very specific language that is called SMILES, but you could imagine of building, and we did actually, something very similar using INCHI code for every single molecule. So it's important overall to be able to capture the large variety of chemical reaction data, both in a summary way, so with a chemical reaction that is representing precursors and product. And it's important in doing so in a way that is directly processable and analyzable by machines. So this is going to be quite a long challenging trip, the one that we are advocating together with other teams around the world, having standardized way of submitting research results in the space of chemical synthesis, whether it's organic or inorganic, having a way to upload experimental protocols that then can be directly processed, extracted, and processed by machine learning algorithms that are learning the chemistry that is embedded in a reaction schema, like the one that you can see here. And of course, I mean, the last slide that I will be showing is not on the model. This is an example, for instance, of the model that we are building for predicting the reactivity. You can use the same data also for building retrosynthetic data, but I want to show you how we use the entire experimental protocol. So I'll just want to give you a perspective of how that is really usable in a context that can help people to narrow down the reaction conditions for the synthesis of new compounds in a more precise way, reducing all the trial and error. And so this is an example, for instance, of another model. This chemical reaction is the one that is described in this paragraph text. So you have a textual description, which is definitely one of the things that is not easy to process by computers. You have as well a schematic representation, and of course you can have a linear representation of this schematic graphical representation of the chemical reaction. And so what we do, the way we use this type of data, is the one of abstracting the text description, so converting this prosaic text description into a concise list of actions, then generalize this list of action into a series of basic instructions where we, instead of referring at the individual chemicals, we refer at the position of the chemical in the paragraph, in the chemical equation. And then what we do, we train a machine learning model that receives as an input, the entire chemical reaction. So the one that is represented here in the upper part, and that is generating as an output the full experimental protocol. So this is really the essence of what we tried to do in the last 3 years, collecting data for building models that simplify and make the work in synthetic laboratories more precise with less errors. I'll stop it here, and if there are questions in the chat, I'm happy to go through them. Thank you so much, and of course, I'm looking forward for discussion and questions.
Excellent. Thank you very much, dear. When you put up that slide of the people involved, I thought that's a stark reminder that these things aren't necessarily just the work of one person, but whole teams, which I think was right. And then, of course, you went on to talk about how that sort of brings chemistry closer to other sectors, which I think was a very interesting observation. And I think the metaphor of sort of atoms and letters, molecules and words, it's the language of chemistry, which we're kind of having to reinvent for machines now. So thank you for that perspective. We're going to move on now to Dr. Yannick Jumbo Funang, who attained a BSc and Masters in the area of computational biology in Germany. And this has led him on to be involved in the development of various semantic and predictive tools covering chemical classification, but metabolite prediction and work with mass spec as well. He's now working with data and cheminformatics at Corteva Agri-Science, and so we're looking forward to the perspective that Yannick can bring, talking to us about what is a chemical from an agrochemical perspective. Over to you, Yannick.
So can you hear me?
Definitely, yeah.
Can you see my screen?
Can see your screen as well, yes.
All right. Hello, everyone. First, I would like to thank the organizers for inviting me today to present on behalf of CODATA AgriScience. One of CODATA's missions is to develop safer and more environmentally friendly crop protection agents. And ideally, such molecules must be effective at extremely low dosage. We're talking about a few grams per hectare of land. They should be really readily degradable in the environment to avoid environmental harm, and they should also be selectively toxic. So this is a fairly complex process, which takes on average about 11 years and $300 million to discover a new chemical crop protection agents and market it. The discovery phase is one of the most challenging phases of the whole crop protection discovery pipeline, which involves hypothesis-driven innovation around the chemistry of interest to design molecules, but also to enhance a number of attributes such as intrinsic efficacy, plant mobility of the pesticide, but also a number of environmental and mammalian safety issues. As the molecule progresses through the various discovery phases, a lot of people or representatives from different functions, such as regulatory, crop protection, discovery department, commercial, field research, supply, are intimately involved in evaluating, refining, improving, or discarding candidate molecules. This is a rather iterative process that consists of several design, make, test, analyze cycles, where a number of molecules are built by exploring and optimizing the structure activity relationships. So as you can imagine, there's a lot of data and chemical information that is generated and ingested throughout the whole process. I'm not to spend so much time here, but these are examples of formats or ways molecules are represented. So we go from the SMARS notation, which is the most popular line notation, with a number of advantages such as readability, interpretability, and a number also of limitations such as the inability to represent 2D and 3D coordinates. Names can be, those are easier ways to represent or to refer to a molecules. They can be used for data mining, but also for the conversion to corresponding structures, and even, for the deciphering structural patterns within the molecules. Formula are very prominent, especially when we are, trying to figure out what the atomic and structure composition of a new metabolite is, transformation products, whether it's in humans or in the water. Fingerprints are also very important for, data, for database search, similarity analysis, and so on and so forth. Chemical families are very important when it comes to grouping the molecules, and they are, an intricate part of data mining, pattern mining, but also mode of action classification. 3D representation can be used for molecular modeling, 3D QSAR, QSPR, and it is important in general to have very efficient and representative graph or language-based representation of molecules, especially graph or sequence-based representation of molecules, as they can be taken advantage of, especially with the purpose of deep learning and representation learning. So, as mentioned earlier, the discovery phase process is a set of iterative design, make, test, and analyze phases or subphases. And candidate molecules are generally, selected or designed upon searching external and internal databases. There is a lot of, there are a lot of processes that can, be involved here. Enumeration of virtual libraries, large-scale screening of databases, visual screening, and also de novo design. And the selected candidates can be further filtered based on predicted or measured physical chemical properties. Other sources of data includes patents, internal reports, and scientific literature in general. And they can be parsed in a semi-automated way, using a combination of various query types, structures, names, chemical families, and other ontological representations, as Lutz mentioned earlier. The results of these searches can be, values such as log D, log P. It can be absolute representation of a chemical space of interest that can be further explored. Now, following the design phase, a selected number of molecules can be synthesized for further test, and it is important to choose the optimal way to synthesize those molecules in order to reduce the speed, the complexity, safety risk, environmental harm. So this can be achieved by retrospective analysis of reaction data from our internal electronic lab notebooks, or by using computer-aided retrosynthetic analysis. So, in that case, reactions can be represented in terms of SMERCs. We have reaction smiles, we have RXN formats, and we also can use physical chemical fingerprints or different type of reaction data, such as yields, to kind of, choose the optimal path and evaluate how safe a specific reaction pathway will be. So in agrochemical discovery, substantial amounts of data is generated by running assays on target species such as, certain fungi, but also non-target species such as bees, mice, and so on and so forth. The data that is obtained are generally captured in a semi-automated fashion and stored in databases. And then cheminformatics workflows and tools can be used for data cleaning and integration into other data storage to facilitate the mining and visualization. It is very important to have these capabilities for the chemists to actually mine the data and come up with some hypotheses. So while we internally generate large amounts of data, we also take advantage of external databases that cover our chemistry and biology of interest when applicable. Now, the analysis phase is a continual process through the DMTA cycle, because the analyses are used in the design phase to help determine, what compound to synthesize. They are used in the test phase to help evaluate and plan tests, and in an oversight role to kind of determine whether to continue a project or not. So in this phase, structural classifications are allowed to partition compounds in groups that can be visualized and analyzed. Predictive modeling is also a really critical component of this phase, as it can allow to design and optimize or filter molecules. The data acquired from tests is usually used along with data acquired from outside or external sources to kind of develop global or local models that can be used later on in the other phases of the cycle. Metabolism and environmental fate prediction tools are very important here in environmental toxicological study. Now, ideally, graph or sequence-based representation shall allow for easy interpretation and prediction of the predictions, and give the chemist a clue of what portions of the chemical structure leads or contribute to a given outcome, whether it's activity or toxicity. So that means we need to have a standard, very expressive representation. Here I provide some an example of prediction explanation using SMILE strings. But it's obvious that SMILE strings, as you know, has its own limitations. So here I have a list of opportunities that this kind of work that we've been presenting here come with. So could we develop a more expressive, standard yet simple line notation that addresses the limitation of SMILE to kind of improve our deep learning methods, representation methods, and facilitate the prediction of, outcome. Can we work together to build and generate high quality data sets? That's a huge issue, and especially in the realm of agrochemistry, because a lot of the data that we see, a lot of the models that we can purchase are usually biased towards the drug space. We have several types of ELN that come with their own formats, and it's difficult to develop parsers that will, parse, each of the ELN formats. So that's also another problem by its own. So the earlier we have like ELN, standard ELN formats and associated parsers, the better it will be for everyone. One thing that is kind of missing, and I know there have been some attempts to solve this issue, is to basically develop a structured IUPAC name converter. There are several applications of IUPAC names, as I mentioned earlier, and this will really help along the way. Improving capabilities for semantic search and reasoning could also enhance data integration, molecular discovery, and so on and so forth. And Lutz and others have been, involved in this space. A model for efficient mathematical representation of structures could also significantly expand the application domain and accuracy of the predictive models. So that's what I wanted to present today. So I would like to thank you again. And if you have further questions, please let me know. Thank you.
Thank you very much, Yannick. I think you raised some very interesting points. I think I was struck early on by how, you pointed out the importance still of some of the old ways that we used to sort of talk about chemicals, including names and formula and stuff like that, and sort of also highlighted the limitations of some of the newer ways and how we need to be able to build on these. But also, I think, how the vast amount of properties that can get generated along the way with that sort of design, make, test sort of like cycle. So thank you very much. Reminder that if you do have questions, drop them into chat. We'll feed them into discussion if they don't get answered specifically there. We're moving now on to Nelson Vinusa, who comes to us from, I believe, at the moment, North Carolina. Is that right? It is indeed. But reading through his bio, quite an interesting background. Looking at design of cancer drugs, forensic chemistry, biofuels, development of new mass spec methods, also involved in initiatives in Ecuador to improve the productivity and research and advance the forensic sciences there, as well as being an associate professor at the Department of Textile Engineering at North Carolina. He's director of the Max Weaver Dye Library, and I think if you see the pictures in his backdrop and on the slide, I think it's a fascinating collection of chemicals there. So looking forward to hearing your perspective, Nelson, on what is a chemical. Over to you.
Thank you so much. You hear me well? Well, I'm going to give a little switch of gears on it because, one of the things that area are tolerance, and we're going to go through dye and pigments and databases from a perspective a little bit on textiles and what we will see. But what you see in the first is, what we're trying to do is try to understand what happened with degradation products in a way, but how we know if we have a dye and pigment. So sometimes, it's important to get some nomenclature really, specific to everyone to understand what we mean by colorant and what's the difference between dye and pigments. But everyone, we consider a colorant, it's something that is developed to absorb or emit light on the visible range, which means 400 to 700 nanometers. But the thing is, one of the important things is these colorants can be inorganic and or organic compounds. Many of us probably in our lifetime we have said, oh, that's a dye, it's a pigment, you just do it interchange these terms. However, for us, when we are talking about this type of compounds, we consider a pigment something that a small particle that you're like in paint. It's insoluble. It will have something that can help you apply it to the surface. Contrary to what we consider a dye, which is something we apply to different substrates, like for example a textile. All of them are going to be kind of in a liquid form, meaning that the dye has to be soluble or partially soluble on the media. So this is important because when we talk about dyes, especially, and everybody is talking about color, everybody of you have any of this, if you have a cotton shirt or a, wool or anything that polyester, you like sports. So one of the things you have to know is depending on what type of fiber you have, we have several type of dyes that will work on it. For example, in cellulosic or cotton, we normally see direct that dye sulfur, but now we do a lot of related to reactive dyes. That means that maybe we actually make a chemical reaction to get together. A lot of synthetic fibers you will find around, especially dispersed dyes. These are hydrophobic in nature. So, you know, all of these depends how they attach to the fabric, and it makes it much easier for us to understand what type of dye and try to see what structure we have. And for us, it's kind of important because sometimes we don't know that. And for everyone that is not as familiar with dyes, I'm going to give a little, see how we see the data and how we can interpret this. So one of the things is if you tick on the, what we call the chromophore, right? In this case, in the nasal, that's going to be there with extension conjugation. That's going to give you something with color. And we can use something we call an isochrome and anti-isochrome to intensify the color or decrease a little bit the intensity, right? So the thing is, depending on the type of fiber you have, for example, a cellulosic fiber, actually we have a covalent bond here. If you have a dispersed dye like a polyester, we see a dispersed dye going into it and trapping in there. So the thing is a mechanical entrapment, there's no interactions. Well, a polyamide fiber will do ionic interactions, and also you will get this. Sometimes we have a nice cotton shirt, t-shirt or pants, you know, you take it to the washer and after a time loses color. Most of the time you will see this as direct dyes. Why? Because normally they are adjusted, you know, in the surface, they're very plain molecule that do hydrogen bonding or dipole-double interactions. So they will not last longer. So that's why you can see reactive dyes will be our favorite way. So we talk about a lot about color and, you know, say where I see, and there's actually a main system we classify this type of dyes, and this is the color index, right? And this is kind of managed by the Society of Dyers and Colorists and the American Association of Textile Chemists and Colorists, the ATCC. There are two different things that we have to remember about the two databases that we have. One is the color index, is the generic name, which technically is going to tell us the application process. For example, if we talk about an acid dye, meaning we are going to have an acid media to apply that dye. Okay, and this is normally going to be like we call acid blue 25. And color index constitution number is the chemical structure related. For example, when we have a monoazo chromophore there, the number from 11,000 to almost 1999 before 20,000, that's going to be monoazo. If we talk about acridines, it's going to be 46,000 to almost 4,700. And inorganic pigments about 77,000 to almost 78. So this is a way for us to see two different ways of information. And sometimes if you are in that chemistry, that's going to be much easier. But if you don't manage this too well, it's going to be complicated sometimes. Why? Because what you see is a table like this. We have a generic name. You know, I can say I want all the ones that are red, and I have acid red 1, 2, 3, 4, and they will tell you the shade, red, bluish red, or red. And also what is important for us, how many commercial products there are about. If you see the CI constitutional number, all are from 13,000 to 18,000. You already, by the slide I said, you see previously, you know all this will be azo. So that way you can understand maybe how's the chemistry. And for me that I have been working in this area, there's a couple of challenges that we are looking at this time. One is color structure can be limited to patterns. So, you know, contrary to a lot of chemicals in many ways, you want to search a dye, it's probably there. If not, it's very hard to get it. So sometimes you cannot correlate these things. And that's why sometimes when I do quality control of dyes that I take from commercial sources, and I try to see what's the structure in the public media, sometimes the math doesn't correlate. So I say, well, there's some changes in the dye, it's still the same name, but they have been changing, and that's because the dyes are synthesized. It can be in different locations, but also they can change just a little bit. The other thing is we have a lack of faster quality control tests. Sometimes we rely a lot on just spectrophotometry. We're trying to introduce a little bit more mass spectrometry, and especially high resolution. one of the things that you will see with this is cost, because normally companies that use colorants, they want to have something that is more accessible. However, one of the things that I find more challenging during this time is toxicological data. And in our first speaker, WorldFAIR mentioned about that. And this is nice to see that we can see something there and get some information about that, because the thing is, sometimes we don't know if the dye can be, there's certain ways we know certain dyes that can be problematic and they're banned right now, but before they were not there. And for example, this is an important, like the Sudan dyes, if you can search it, if you Google it from them. The thing is, these dyes are still being used as to, enhance color in food, in spices, right? Or they're still using cosmetics, so they can become a problem if we ingest it or so. So the thing is, there's still things on there. Plus, we talk about dyes, but not under the derivative, so they come up the treatment, what we do. So to do that, I've been fortunate to be part of the Max Weber Dye Library and directing, and that's why we say enhancing databases. What I mean by this is we have the Weber collection that you can see is almost 100,000 dyes that you can see from there. And important thing here is that we try to combine this to other accessible things for dyes, because these are molecular structures that can be useful for us. And you can have color structure relationship, fundamental studies, spectroscopy, try to do a lot with those. Importantly, how unique they are. On a paper we published in 2017, we gave 150 structures to all the scientific community together. And we compared those 150 structures to 58 million at that time in 2017. What we found, 143 structures were unique. So that means, that day we can buy lottery and try to get some good luck at that time. So what we're going to do, we're digitalizing the library. We have around 5,000 and we try to get to 20,000 soon. We're going to share with this cheminformatics with, now it's going to be nice because we can use several tools to explain that and be able to work on different areas for us, and also enhance STEM education. If you have interest on it, this is the Weber. It's an open access that we publish the analysis if it's there. With that, because due of time, I want to thank all my team, and of course NIST, that we're working with them, and Thermo Fisher to send all the databases. You can follow us on Twitter. And with that, one of the things we do is we can have a dendrogram that we can classify based on molecular weight. So we can see dyes in a different way. With that, I will try to wrap up. Thank you.
Thank you very much, Nelson. That was fascinating. I think it was good to be reminded at the start there that not all the chemicals we deal with are organic. We need to think about inorganics too. I think it was interesting that you have to think not just about the substances themselves, but what they're interacting with and how important that is. And then that sort of need to sort of look to various different data sources to get that toxicology information or other properties that might be important to really advancing that. So thank you very much for that insight. Okay, our next speaker is going to take us back into the territory of drugs, I think, certainly the pharmaceutical sciences. So Nick Lynch has many years of experience in research and development in the pharma industry, covering early stage discovery through to late stage health informatics. He's been involved in his career in various pre-competitive initiatives, most notably, I would say, the Pistoia Alliance, and being a sort of early instigator of that project, and also various IMI projects. He now operates through Curlew Research, has quite a good grasp of the challenges associated with data interoperability. So looking forward to hearing his perspective on FAIR data in the life sciences from a chemistry perspective. So over to you, Nick.
Thanks very much, Ian, and thanks very much for the chance to talk about some of our work and obviously the work of lots of other people too. So one way, we've heard a lot of great talks already and some really great overlap, I think, with Yannick's and some of the other talks before, in the sense of the data, the entities that we're dealing with, and also the data. And one way we like to think about the life science world is that in any data that we create, it's a long tail. This sort of picture of a comet is one analogy we like to use, in that you've got the the process of creating the data and planning to create the data, and we'll come to that a little bit in a minute. But then its real value is its long tail, partly because we never know everything at any one point in life science, and we want to build on data, remix data, bring in public data, just as Ian mentioned with the crystallographic service, with protein data from from the PDB, and with wider data. So we always have to think about the wider aspects and the interoperability of data, and this is particularly where FAIR data is helping us bring data together in the right way so that we can make better decisions, not just for ourselves, but with more of the automated tooling that we have access to, and obviously hoping to use more of AI and ML. I mean, although we talk about chemistry, I'd like to make the point that even with the richness now of the potential drug modalities, many of them, or a large percent of them, involve chemical structures, even for the larger sort of RNA and other vaccines that have become to the fore in the last 2 years with COVID, that we can represent them with a chemical structure. And so although small molecule research perhaps has been to the fore in the past, all of this richness of chemistry needs to be managed appropriately, as well as, more importantly, the associated data with those entities. So even if you're working on an oligonucleotide, you could hopefully represent it as a monomer using the right chemical chemical sort of nomenclature, as well as then all the other associated data. So it's really important that we consider not just the chemical part, but all the other associated assay data, regulatory data, and so on. So we've heard a little bit about this from Yannick, and in a way we could have probably changed talks, but this whole concept of the data life cycle. So in the sense of design, make, test, or we've expanded it a little bit here, and I wanted to pick out where FAIR data can really help here. So here we've got the design phase where you're trying to learn from all the previous data that could have been generated within your company or externally. And the key here, I think, is about data being born FAIR, to make sure that when data is coming to life, that we're using the right identifiers, we're appropriately using the right ontologies. And this applies not just to the point of working with the chemical or the asset in the beginning, but all the other associated data that you might collect on that asset. And we heard earlier on about the use of knowledge graphs and other data warehouses, because without that capture of data in an appropriate format at the beginning of the life cycle here, we have no hope of really making value from it at the reuse point. And there are some key things that FAIR data can really help with, and what we see with our work with customers is the use of a data catalog, for instance, to help us secure data, as well as the importance of interoperability. Even if we're using one ontology or vocabulary, how can we map that to another ontology or in a similar space? Because we'll never be able to necessarily speak the same language, but if we can translate between our languages, then we've sort of made half the battle successful. And again, it comes back to this findability and accessibility, which is critical. Within the sort of life science world, we have a number of challenges in the sense that research never happens in one place. We're working with collaborators and research organizations. So we have to be able to manage this interoperability when it comes to sharing data. We work with other public-private initiatives too. We also have to think about the importance of maintaining stability in our identifiers when we migrate systems. And a particular theme is when companies come together, how can we manage the interoperability of two companies with two different systems? We have to think that's a real need, and we see a lot of that even now when companies want to work and collaborate, of being able to speak the common language in their shared and mixed data sets. And I think one journey that we think of about FAIR is that often you'll want to work with FAIR locally on your data. And then you sort of have to think about these onion rings as you grow your interest levels from your data to company-wide and so on, until the point where perhaps you're publishing your data or working with a global data set. And all of those things have to be considered in terms of the data, not just the chemical itself or the chemistry itself, but all the other experimental assay, screening, and other related omics data that you will collect. And so again, it comes back to this long tail of data that we need to work with. There's lots of efforts to help us with this, both with FAIR Plus, which is an IMI, IHA initiative, and many other initiatives mentioned here that are all working on helping us make our data interoperable. And I think that's going to be a key message for work for all of us, as we both want to use the data appropriately, as well as making the most of ML, because at the heart of good ML and AI is good data. And the more that we can do to make the data born fair at the start will hopefully help us with those our goals and objectives through research. I'll stop there and happy to take questions and comments now or later.
Thank you very much, Nick. So yeah, we'll hold questions over to the discussion session afterwards. But I think, yeah, you've raised a point that was hovering in my mind throughout these, particularly around the concept of data being born fair. I think we heard in some of the earlier talks how people were trying to sort of retrospectively get information out. But yeah, heads up to the speakers here is one of the areas I want to sort of explore a bit in the panel discussion is what are the challenges and opportunities of thinking about how to make data fair from the off rather than having to retrospectively tease things out. And I think thank you too for sort of reminding us about the sort of realities of the corporate world when companies merge and working with collaborators, and the importance of having, you know, well described data for enabling those collaborations to be effective. So thank you. We've heard a couple of times throughout various talks the importance of things like toxicology and making sure things are safe for the environment. So our final speaker, Gunther Shadow, comes to us from a regulatory background, which is relevant here, of course. He's led a significant number of standards initiatives relating to healthcare and medicinal products in this space, and has been involved in IUPAC divisions. He's an associate professor of medical informatics at Indiana University, and now is president of Pragmatic Data LLC, who offer innovative software solutions that help healthcare and pharmaceutical industries in the context of satisfying regulatory requirements. So Gunther, good to have you along and look forward to your perspective.
Oh, thanks for that slick introduction. Okay, let me just go and share my screen, share, and go to the slide. And here we go, IUPAC FAIR and there. All right, so yeah, you mentioned environment. I was briefly, I was related to something in pesticides also through an FDA, EPA collaboration, but that fizzled out at some point, long time ago. Yeah, I started with the FDA about 2006 or no, 2004, 3, something like this. And my company were providing the software and some assistance to the worldwide pharmaceutical industry that they have to make certain post-marketing submissions to the FDA using this SPL standard, which is also quite capable to represent chemical data, chemical substance definitions. And recently we've also worked with the NCI on extending that for reactions. But so I'm starting this, I was actually quite intrigued by the question that is, what is a chemical? And there's actually an answer that you might not like to hear. So the ICH, that's the International Conference for Harmonization, and harmonization means here between regulators. So the industry and the regulators sit at the table and talk about how they can harmonize regulatory processes. They have come up, it's also 10 years ago now, over 10 years, with a of medicinal products. And one of those standards of the set of standards was called ISO 11283, and that is for the description of chemical substances, or I shouldn't say chemical substances, I should say substances, And they define a chemical, right? Basically it's a small molecule. Here's text, you can read it yourself. But basically they say chemical is a small molecule, and as opposed to a protein, nucleic acid, random repeat polymers, and also the very important area of structurally diverse substances, which is basically organically derived, not organic in terms of organic chemistry, but from organisms derived, right? Such as a lot of products that we get to see actually are allergenic extracts, which are all this sort of thing. But then, of course, this whole area of dietary supplements, which are not very much regulated, are in this space. So now, as far as this picture here, I brought in because I was interested in understanding what is the how many molecular entities, new molecular entities, are, how are they? What's the importance compared to biologics, which are mostly these biologics are sequences. They're mostly monoclonal antibodies, and so it was interesting for me to see that really the small molecules are still a very important area, even though people like to speak about more complex molecules. But it seems like the small molecules are still quite an important area in current development of therapeutic substances. So this text here comes from this ICH-M5 module 3. Now, what is module 3? There is one of the things that the ICH did is they standardized the application package. That was also called the Common Technical Document, and that has an electronic form, and it's then called the ECTD, the Electronic Common Technical Document. And that's basically a large collection of documents, and the standardization of the table of contents is what these people do between where the industry and the regulators are coming up with how should we structure an application which can well be thousands and tens of thousands of pages that are being submitted in the application phase, which summarize all the clinical studies, of course, that have happened, non-clinical studies. And as you can see here, these modules that are involved: module 1, administrative information summary, and then comes this whole area, module 3, quality, and then module 5, non-clinical, and module 4 and 5 is non-clinical and clinical studies. And so interestingly, this entire module 3, quality, is where the chemical questions lie. So there's a division between drug substances, so substances, and products. And I guess I can drill a little bit more down. In the drug and the substances, they deal with nomenclature, the structure, general properties of the substance, how they manufacture it, then how the substances can be characterized, and impurities are obviously a big deal. So the quality standards that are described with specifications, and then batch analyses, and then you have to show that your whatever heavy metal amount is below a threshold, and your impurities and solvents and whatever else is remaining from the manufacturing process is under a certain threshold, and so on. And then the other side is the drug products, which of course beyond the chemical to how you a dose form, which is quite interesting. And what is interesting is also the overlap between maybe complex substances. We hear some in INCHI, for example. I've been attending several INCHI events because we use INCHI as a core building block for the specification of complex substances in our SPL substance specification world. And so I go to INCHI meetings. I hear what people are developing in terms of new INCHI variants or modalities, and deal with nanomaterials and all sorts of things where you have basically just just above the molecular scale, a description of how the substances are put together in space and beyond the simple bonds or in proteins, the tertiary structures and so forth. So there is an interesting overlap between what's a substance and maybe what's a specific form of the substance. But by and large, it's distinguished in this in the thinking of the application package. You know, there is the substance description on the one hand, and then the dose forms on the other hand. One thing that I sort of dwell on this because it continues ringing in my mind when I had we wrote recently an article about a paper about using the SPL standard for reaction for description of reactions, and they are talking with various synthetic chemists. They were very adamant that actually the form of a substance is quite important in terms of how you can use it for maybe automated driven reactions. So form apart, you know, aside, in addition to substance, is quite an important aspect. Although when you ask for what's a chemical, well, then you would deal with the substance. And to maybe I should skip to FAIR principles. What I'm interested in is maybe not so much. I've been dealing with omics data and stuff like this, but since I'm not actually in the in academia anymore, but you know, I decided to just go full time on my development on software and so forth. What I'm interested in is making data available, you know, on just basic resources. So for example, in terms of substance definitions, where most of the people get their definitions of what's the substance, there are these nomenclatures, like for example, the international the INN, the committee for the assigned established names that are being given for generic names to new molecules. So they publish these books, articles, and maybe recently they started electronic publishing, but it's actually it's still documents, mostly text, some pictures. And what I'm interested in is, and that's what this SPL standard does very well, is it allows you to take the text to continue with writing documents, because that's what that whole industry does. And that's what nomenclature committee do, and pharmacopoeia. They are all books, they're all documents with monographs that have a lot of text that may use any kind of free form pictures, but then they have structured data also. So what this SPL standard that I have built and implemented several times is quite useful, is that it keeps the document together, which is, in my view, it is a fair issue, because it makes the data accessible. Oftentimes when you have, let's say you have a super great database that has millions of records and is certainly very interesting, there's a bit of a problem to actually understand what's going on. And when you have highly curated data, which is what we deal with in the pharmaceutical world after the drug discovery. So when you go down into the application phase, everything is very highly curated. You're not speaking about some totally unknown or millions of substances anymore. You're speaking about highly curated things. So there's monographs, there's things written on them, there are essays and all of that. And unfortunately, this information is partially public. Much of it stays sort of hidden under what is it? It's confidential, right? So unfortunately, we cannot see the entire quality standard package that the industry sends to the FDA. We cannot see this, but what we can see, so pharmacopoeias or nomenclature. And substance definitions out of such things that the FDA has been running the substance information system, and now they have been publishing another. They have been setting up something along with the Europeans, which is the GSRS registration system, and we've created an SPL version of that. So in these highly curated areas, I believe that what makes data most accessible is that you keep the document context, and that you disseminate this data on places where people can go and get it, which the FDA has been doing with drug product data and substance data, and to in the case of the substances, we have submitted these to PubChem, and they got integrated into PubChem. But there is a repository of all these documents where you can go and get them. And it's not perfect. I would like to actually make this much better than it actually is. The findability is a big deal, but the accessibility in documents, that to me is very important. And I actually almost I several times thought about we should sit down in IUPAC and actually make standardization of such monograph documents, so that these monographs would be issued in more fair ways to be then interoperable and reusable, because that's what you need, right? If you just deal with the text, there's a lot of text mining going on in this field, as maybe the people who work with Google or other places can tell. There's a lot of text mining going on, or structure recognition from pictures, but I think we can do much better with this if there was a common rich document standard which we can contribute.
Günther, thank you for that very much indeed. And I think that's taken me literally to the question that I want to quickly go around the panel. We've only got about 5 or so minutes. So I think first, thank you for reminding us about this sort of like nuances around, it's not just molecules we need to represent, but substances, products, all that sort of thing, and mixtures. But I think what you've been talking about, that reporting thing, is the question I want to throw out there is just to give each speaker an opportunity just to say 1 or 2 things about how we should be doing things differently, so that it is easier to take advantage of the data that is available, and makes it easier for people to make data available in order to really sort of take advantage of AI, machine learning, and just do better science. If you also want to think about how an organization such as IUPAC might be able to help with this, or other organizations that we should be bringing to this conversation, that would be really great. And so we'll go in the order that the speakers went, give people a chance to say something. So Lutz, first of all, any observations around that theme from you?
Yeah, I have one point which is very clear, and which is also a point where the IUPAC could help. And this is really in the field of organometallic compounds. So as we know right now, the INCHI does not represent coordinative bonds, hydrogen bonds, or things like that. So we have a discrepancy between the expressivity of the different structure representations. As Yannick already said, he would like to have a more expressive SMILES thing, which would directly lead to a more expressive INCHI. So in mole files, we can have coordinative bonds and hydrogen bonds. In SMILES, we can have quadruple bonds, which we can't have in mole files. So there's a discrepancy of the expressivity of the structure representations, and it would be great to have one common, standardized, commonly agreed expressive structure representation. So that's my point.
I think, Ian, you are muted.
I was asking your perspective, Theo, on sort of the challenges you face getting information out of all these diverse sources. How should we be doing this differently? And what's one of the key things that might help move the needle here?
Yeah, absolutely. I want to amplify what Lutz was mentioning. Clearly, we do have lots of challenges in molecular representation, but on the other hand, another thing that we should do as a IUPAC is really okay, I'll open a little bit of a parenthesis and try to be short and maybe a little bit popular. At the end, we are dealing with human creating content. And we have been experiencing a lot, I mean, that putting very rigid structures not necessarily leads to a successful adoption. So I'm a strong supporter of the fact that we do have quite some technology, for instance, related to what I was showing in the space of processing natural language for structuring data that is reported in a very chaotic way by people. At the end, the real use of machine learning technology is actually the one of creating a little bit of order where there is chaos. And so I think as a IUPAC, probably we should explore and adopt more technology that are trying to bridge. I mean, not necessarily tell people how they should structure the data, but trying to make the work of people using the data easier while letting the freedom to who is generating the data. That's it.
Brilliant. Thank you very much. Yannick, you mentioned not just structure representation, but also properties as well. So any kind of reflections that you have on this area?
Well, often people in the industry, I mean, they're generally concerned about sharing certain type of information. And in a previous discussion, I think last year or so, there was a question of how can we represent data and share data without necessarily sharing structures. Sometimes even for structures that are no longer, of interest within the company for a specific project, we can make a case that they might, be important in another project, through retrospective analysis. But there are maybe abstract ways like physical chemical properties to, provide a description of the molecule, with respect to, the log P, constitutional descriptors and things like that, without necessarily conveying what the structure is. And that will help sometimes at least develop models that can be reused by the whole community without having to describe exactly what the structure of the molecule is. When it comes to actual representation, if you look at the literature, you see that SMILES is still very popular, of course, but there are derivatives of smarts like selfies and big smiles and all that can be used for machine learning and reinforcement, representation learning, in order to design models that can allow to really specify the nuances within different molecules. I think one advantage of SMILES is that you can randomize it. But a disadvantage of it is that it's not necessarily providing a standard way of canonicalizing it. So come up with something that is expressive enough that will provide maybe not just the covalent interaction, the description of covalent interaction, but also to some extent non-covalent interaction will be interesting. I don't know if it's a kind of a SMILE, attached to some other, sequence-based description, because I'm thinking, representation of the molecule, but also the usage, the practicality for, downstream applications like QSAR modeling, things like that. Because, the limitation of the SMILES, sorry for not showing my face here, the limitation of the SMILES is that once you get to certain really complex molecular properties, having only a description of the covalent bond is not enough. And doesn't matter what type of algorithm you use, you're going to be limited. And given that we don't have that much data, the size of labeled data sets is generally small. The applicability domains that you get from this model is also fairly small. So having something that is really expressive and easy to parse will be fantastic.
That's some great suggestions there. And I think I'll just add to the sort of first point there about it's not just sharing positive data, but also getting hold of negative data, I think is becoming increasingly important for training models. And yeah, the idea of going beyond covalent bonds, I think, definitely resonates from a crystallographic perspective too. Nelson, yeah, what's going to help you? Maybe, I'm not sure if it's fair to say play catch up, but I think kind of it sounds like there's a lot more challenges, sort of like just getting up to speed with some of these other sectors we've heard about. So what would make the biggest difference?
I think one of the great things about it is we think, my perspective has been always to think that dyes as organic molecules or inorganic molecules. So how they can go to transition to other areas, and that's what my push is on making this dye library more accessible. Hopefully in a couple of 1 or 2 years, we can have probably the 100,000 dyes available for researchers and all that. So one of the things that I like is, on the study we did, we just make some things with the Lampiski rules, and we see that, a lot of dyes follow those. So you can see how this can be going to another transition there. Plus, the other thing is other areas like imaging or so how to get the properties of things that are there, can help. And, one of the things that makes you think like that, when we did that statistics, like we give to the scientific community 150 dyes, and only 7 were already on the database on ChemSpider at that time. You can see is like how unique this dye library is, because sometimes with other speakers, like there are things in companies that, and by the way, this dye library comes from Eastman Kodak at that time with Eastman now. It's a collection from late 50s to the almost 1989 that we have seen tonight. So there are dyes that probably were there that not many people use it, and how we can contribute to make it something different. And what we have found is data curation it can be challenging, because some of the things on the search, some of the dyes maybe have salts, so you have to make sure that they are clear and all that. So it just depends where you're going to be searching and what all of them you do. But ideally, what we're trying to do is make these molecules accessible for every field that it can be available. I think that's something we do. And
yeah.
It's challenging with the structures because everything is patented on some of the dyes, the new ones. But I think that's it gets time to do it. But I think the dye library, the Weaver library, can be a great catalyst for a lot of things.
Thank you for that. I think it's a reminder that in some sectors we've got to get to that point where it's that 5 stars of open data. The first thing is get it out there so people can start taking advantage of it, etc. So thank you. We're running very close to time. So Nick, in 30 seconds, are you able to just give us a nugget of wisdom here?
Yeah, I think two words of things in terms of sharing best practice. We've heard a lot about trying to extend our standards, but I think we have to try and make the most of what we've got. So if we can actually define what the boundaries are for the representations, I think that's going to help both for human work and, even more importantly, the machine readable aspect. And so I think we should, be careful about trying to find the perfect standard, because we're never going to, because in industry, and I believe in academia, it's about getting the value from what we do and making sure that we can obviously to deliver to our different patients and other customers.
Okay, thank you for that reminder as well. And Gunther, is there anything that you feel hasn't been covered in 20 seconds?
Aha, I get even less. Well, to me, the most important points are the low technical infrastructure requirements. You can't overburden people with a huge amount of technical complexity for them to publish their information. You also shouldn't constrain them too much by, let's say, you build a big data, a very detailed data model, and then you provide some spaces where they can put comments. You need to give them the freedom to say what they want, and then allow them to mark up things that are probably of shareable interest. Also, coming from the other side, limitation of the scope of standards. INCHI is a wonderful thing. And we use it to a very large, very deep extent. We use INCHI, but I don't think INCHI should be extended to make ever more complicated superstructures with INCHI. It's much better to use these INCHI components and then use a more easily composable structures like XML, very good for documents, and build more complicated things up from there, rather than having each standard try to do everything under the sun.
Brilliant, very concisely put and very on point, I think. To those that are still here, thank you for staying. To those who might have to go, we understand, but I think for those who can stay, Fatima probably got a few closing words.
Thank you, Ian. I know we are a little bit out of above 90 minutes so far. So let me share a couple of screen with you here. And I think we do have a poll that we really hope that you can have your feedback a little bit here. Let me just try to launch it. All right, so while this poll is going on, we really would like to have your feedback, what we can do next and how your satisfaction about this event. Perhaps we can share with you what's coming next. And we're going to be there in so many events, continuing this webinar series next month or the early December with the coordination with ICS Sense. And then we will have early prototypes by the beginning of the next year. Next year, spring, we will be having so many technical workshops regarding our deliverables and our services that we are trying to develop in this project. So in spring, we have virtual workshops coming, and then we have in-person workshop in the ACS Indianapolis in March. In summer, we will be also organizing a symposia in the IUPAC chains in Netherlands, and also a Senf Data Summit in the ACS San Francisco. Coming in winter, we will be there in the International Data Week in Salzburg. So stay tuned, get engaged. We want your feedback in our services that we are developing. Your engagement is very highly appreciated. So that is what we are trying to let you know about. So our this webinar will be recorded and uploaded in the YouTube channel of the IUPAC as the previous webinar as well. So if you want to rewatch this, you can find it very soon. Our next webinar and final webinar of this series will be co-hosted by the ACS Senf and will be about the status and the future of developing digital machine readable depictions. So stay tuned to know about that. And if you want to contact us, you can follow up on Twitter and follow up the IUPAC on Facebook, and as well as emailing us. Lastly, I would like to let you know about an event that the IUPAC is organizing since 4 years right now. It's the Global Women Breakfast to empower women in chemistry. And now this year, 2023, all disciplines are invited to organize independently their events. Last year, we had around 400 breakfasts in around 80 countries across the globe. So if you wish to have more discussions about empowering women in chemistry, everybody is invited to do that. So lastly, I would like to thank all of you for attending, for our amazing speakers for providing their insight, and this is an open invitation for you as well to be engaged in our upcoming event. Thank you, everyone. Thank you.
Thank you, Fatima, and echo my thanks to the speakers too. Thank you.
Thank you.
Thank you.
Yeah, thanks a lot, everybody.
Thank you.
I learned a lot.